Good morning, friends. We are studying microbial metabolism and we studied different pathways in microbial metabolism topic. We also discussed how ATP is generated during the electron transportation. We studied electron transport chain and the sequence of electron transport carriers in that particular chain, the arrangement of the electron transport carriers that in sequential manner. Depending upon the oxidation reduction potential, the carriers are arranged depending upon the electronegativity, and these electron carriers, they are carrying electrons into them, and ultimately the electrons will be accepted and protons are accepted by molecular oxygen in aerobic respiration. So we know electron transport carriers including NAD, FAD, flavoproteins, ferridoxin, coenzyme Q, as well as chain of cytochromes. They are arranged in specific manner. And electrons and protons are transported through the carriers. Ultimately, at 3 steps, energy in the form of ATP is generated. Now we are studying here the chemoosmotic model, which is proposed by Peter Michael, and this model is explaining how ATP is generated during transportation of electron. The chemoosmotic model was proposed by Peter Michael to explain synthesis of ATP by using energy release by electrochemical gradient of proton. What happens whenever the reduction reaction is there? From that reduced substrate, the electrons and protons will be transferred to the electron carrier and the substance itself get oxidized. Whenever the NAD is the primary acceptor of protons and electrons, NAD will be reduced to NADH. Ultimately, these protons and electrons will be transported through the electron transport carriers sequentially and then accepted by final electron acceptor. So this is the way in which the electrons are flowing. The electrons flow through the carriers, electron transport carriers. So Peter Michael suggested that the synthesis of ATP occurs during the transportation at 3 steps, but this energy is released by electrochemical gradient of proton. Gradient of proton means that at one side of the cell membrane, the concentration of proton is high, and inside the membrane, the concentration of the proton will be low. So whenever there is a concentration high or high concentration and low concentration across the cell membrane, this will create a gradient. Gradient means high concentration of proton at one side of the membrane and low concentration at another side of the membrane. So this is known as concentration gradient. So during the flow of electrons, the protons will be expelled out, and due to this protons, they will get accumulated at one side of the membrane or outside the cell membrane. So this will create high concentration of the protons outside the membrane, and as a result of this, inside the membrane, what is the situation here? The protons concentration will be low, so it means that the concentration gradient will be created across the membrane. High concentration outside the membrane, low concentration inside the membrane. So this is known as proton gradient. So this model suggested that the special enzyme ATP synthase works as proton channel and harness the energy from proton flow to bring about the synthesis of ATP. Here the enzyme play very important role in synthesis of ATP. The enzyme is ATP synthase. So ATP synthase, it will harness the energy and it works as a proton channel. Works as a proton channel means what? The ATP synthase enzyme, where it is located in the membrane, through this location, through this site, the protons try to re-enter inside the cell. Means whatever protons are expelled outside the membrane, these protons will try to re-enter inside the cell, and this entry point is nothing but it is the location of the ATP synthase enzyme. So where the synthase enzyme is located, through this site, the expelled protons will be try to enter inside the cell, and the energy from electron flow is used to create the proton gradient in periplasmic space. Periplasmic space means the space between cell wall and cell membrane. that is known as periplasmic space. So electron flow, whatever electron flow is there, that is utilized to create the proton gradient. What is proton gradient? Again I am explaining here. The proton gradient means the concentration of proton is variable. Outside the cell membrane, proton concentration will be high. Inside the membrane, proton concentration will be low. So this is the gradient. This is called as gradient. So across the membrane or in the periplasmic space, you will find high concentration of proton at one side, low concentration of proton at other side. So this is known as proton gradient. So proton pumping not only create the gradient but also create charge difference. Proton pumping in the sense the flow of protons. So this flow of proton not only create the gradient but also it create the charge difference, means high density charge at one side and low density charge at another side. So it means that whenever the protons are expelled outside the membrane, the high concentration of the proton and high charge density will be there across the membrane or outside the membrane, and low concentration of proton inside the membrane, that is the low density gradient inside the membrane. So this is the concentration gradient. So this negative charge is created at inside of the membrane and positive charge outside the membrane. That's why I am explaining this one. The same thing I explained. निगेटिव्ह चार्ज जो आहे तो आतल्या साईडला जास्त होतो आणि म्हणजे सेलच्या आतल्या बाजूला, आणि सेलच्या बाहेरच्या बाजूला पॉझिटिव्ह चार्ज पार्टिकल जास्त त्या ठिकाणी ॲक्युमुलेट होतात. सो ॲट द आउटर सरफेस पॉझिटिव्ह चार्ज विल बी मोर अँड इनसाइड द सेल मेम्ब्रेन निगेटिव्ह चार्ज विल बी मोर. म्हणजे एका बाजूला पॉझिटिव्ह चार्ज जास्त, एका बाजूला निगेटिव्ह चार्ज पार्टिकल जास्त, म्हणजे ग्रेडियंट क्रिएट होतो. सो व्हॉट इज द एक्झॅक्ट मेकॅनिझम हिअर? द कॉन्सन्ट्रेशन डिफरन्स अँड द चार्ज डिफरन्स. कॉन्सन्ट्रेशन डिफरन्स म्हणजे एका बाजूला जास्त कॉन्सन्ट्रेशन प्रोटॉनचं, एका बाजूला कमी कॉन्सन्ट्रेशन, ह्याला म्हणायचं कॉन्सन्ट्रेशन डिफरन्स. चार्ज डिफरन्स म्हणजे एका बाजूला जास्त चार्ज असलेले प्रोटॉन संख्येने जास्त, एका बाजूला कमी प्रोटॉन चार्ज. सो ड्यू टू पंपिंग ऑफ प्रोटॉन इज क्रिएटेड अँड दॅट इज कॉल्ड एज प्रोटॉन मोटिव्ह फोर्स. म्हणजे हा जो चार्ज डिफरन्स आहे किंवा कॉन्सन्ट्रेशन डिफरन्स आहे प्रोटॉनचा, तर त्यामुळं काय निर्माण होतो? प्रोटॉन मोटिव्ह फोर्स निर्माण होतो, किंवा त्याला इलेक्ट्रोकेमिकल फोर्स म्हणायचं, किंवा PMF शॉर्ट फॉर्ममध्ये. सो PMF दॅट इज फॉर प्रोटॉन मोटिव्ह फोर्स. दिस इज ड्यू टू पंपिंग ऑफ प्रोटॉन्स टू द मेम्ब्रेन. दिस विल क्रिएट द हाय चार्ज अँड लो चार्ज density particles across the membrane, so that the gradient will be created here. At one side of the membrane high concentration, on the side low concentration. Similarly, charge density also low charge and high charge particles. So due to concentration difference, proton tends to move from high concentration to low. आता हाय कॉन्सन्ट्रेशन कुठल्या बाजूला आहे? बाहेरच्या बाजूला, सेलच्या. तिथून लो कॉन्सन्ट्रेशनकडे, म्हणजे लो कॉन्सन्ट्रेशन कुठे आहे? सेलच्या आतल्या बाजूला. तर हाय कॉन्सन्ट्रेशनकडचे जे प्रोटॉन्स आहेत ते लो कॉन्सन्ट्रेशनकडे फ्लो व्हायला लागतात. सो द फ्लो ऑफ इलेक्ट्रॉन्स इज फ्रॉम हायर कॉन्सन्ट्रेशन टू लोअर कॉन्सन्ट्रेशन विथ रिलीज ऑफ एनर्जी. मग असा फ्लो होत असताना एनर्जी रिलीज होत असते. दिस एनर्जी कॅन बी यूज टू फॉर्म ATP. दिस एनर्जी इट इज युटिलाइज टू सिंथेसाइज ATP. ही जी एनर्जी त्या ठिकाणी बाहेर पडते ती एनर्जी ATP सिंथेसिससाठी किंवा ATPसाठी वापरली जाते. The flow of 3 to 4 protons generate one ATP molecule. Means what? Whenever there is a flow of protons, number will be 3 to 4. 3 ते 4 प्रोटॉन जेव्हा मूव होतात किंवा फ्लो होतात या साईडकडून, हाय कॉन्सन्ट्रेशनकडून त्या साईडकडे म्हणजे लो कॉन्सन्ट्रेशनकडे, तर त्यावेळेला एक ATP मॉलिक्यूल तयार होतो. सो वन ATP मॉलिक्यूल विल बी फॉर्म whenever there is a flow of electrons from high concentration to low concentration. Whenever 3 or 4 proton will move from high concentration to low concentration, per flow one ATP molecule will be produced. So what is the mechanism of ATP in case of anaerobic microorganism? Anaerobic means the respiration of organism in absence of oxygen. ऑक्सिजन नसेल त्यावेळेला जे ऑर्गॅनिझम त्या ठिकाणी सर्वाईव्ह होतात किंवा ऑक्सिजन लागत नाही ज्यांना, त्या ऑर्गॅनिझमना अनएरोबिक रेस्पिरेशन किंवा अनएरोबिक ऑर्गॅनिझम म्हणतो. सो अनएरोबिक रेस्पिरेशन is there in some organism we observed. It is carried out by anaerobic organism which does not use oxygen. They do not use oxygen for their respiration. They do not require oxygen for their respiration. So they are using inorganic molecules other than oxygen. So who is the final electron acceptor in anaerobic microorganism? In aerobic organism we know electron acceptor will be oxygen. In anaerobic microorganism other than oxygen will be the final electron acceptor. It may be carbonate, it may be sulfate, it may be nitrate, like that. The inorganic molecules like nitrate, Fe2, sulfate, carbonate and H plus ions can be used as a terminal electron acceptor. Even protons can be also used as a final electron acceptor. In E. coli reduces nitrates to nitrate. E. coli मध्ये nitrateचं रिडक्शन नायट्राईटमध्ये होतं म्हणजे NO3 to NO2. Pseudomonas species reduces nitrate to nitrogen. Pseudomonas they are going to reduce nitrate to nitrogen gas म्हणजे NO3 to NO2, NO2 to nitrogen gas. Methanogenous reduce carbonate to methane. मिथॅनोजेनिक ऑर्गॅनिझममध्ये कार्बोनेटचं रिडक्शन म्हणजे CO3चं रिडक्शन होऊन मिथेन गॅस तयार होतो म्हणजे CH4. So here some examples of anaerobic organism has been given. In case of methanogens, carbonate reduces to methane, Pseudomonas reduces nitrate to nitrogen, and E. coli reduces nitrates to nitrites. These are some examples of anaerobic. The proton motive force generated during electron transport is used to form ATP. Whatever proton motive force is created, proton motive force means flow of electron, flow of protons and electrons you can say through the electron transport carriers, and protons, high concentration of protons will be generated outside the membrane, low concentration of proton will be generated inside the membrane. So this will create the proton motive force. So this force is utilized to form ATP, and the amount of ATP formed in anaerobic respiration is lower than aerobic respiration because of partial operation of TCA cycle. So here we observe that TCA cycle mainly operates in aerobic organism, but there is a partial operation of TCA cycle in anaerobic organism. So whatever the amount of ATP is formed in anaerobic organism, that is because of partial conduction of the TCA cycle. थोडं TCA cycle partially anaerobic organismमध्ये कंडक्ट होत असतं, ऑपरेट होत असतं, त्याच्यामुळे त्या ठिकाणी ATP किंवा हे तयार होतं, एनर्जी तयार होते. द फॅकल्टेटिव्ह अनएरोबिक ऑर्गॅनिझम स्विच बिटवीन एरोबिक अँड अनएरोबिक रेस्पिरेशन अकॉर्डिंग टू एन्व्हायरमेंटल कंडिशन. Facultative anaerobics means they require low amount of oxygen. That means they can respire, they can survive even though oxygen concentration is less. So in those organisms, in between aerobic and anaerobic respiration, there is a such type of the environmental condition, and due to which the energy will be produced. If oxygen is present in the environment, they will prefer aerobic by replacing the gene required for अनएरोबिक रेस्पिरेशन. म्हणजे स्ट्रिक्टली अनएरोबिक आणि फॅकल्टेटिव्ह अनएरोबिक याच्यामध्ये फरक आहे. स्ट्रिक्टली अनएरोबिक ऑर्गॅनिझमना अजिबात ऑक्सिजन चालत नाही, पण फॅकल्टेटिव्ह अनएरोबिक ऑर्गॅनिझमला थोडा ऑक्सिजन असला तरी चालतो. जर समजा ऑक्सिजन त्या ठिकाणी एन्व्हायरमेंटमध्ये प्रेझेंट असेल तर फॅकल्टेटिव्ह अनएरोबिक ऑर्गॅनिझम दे विल प्रेफर एरोबिक रेस्पिरेशन. तर फॅकल्टेटिव्ह अनएरोबिक ऑर्गॅनिझम काय करतात, एरोबिक रेस्पिरेशन प्रेफर करतात. हाऊ they replace the gene required for anaerobic respiration. अनएरोबिक रेस्पिरेशनसाठी जे जीन्स आहेत ते जीन्स एक्सप्रेस होऊ न देता ते एरोबिक रेस्पिरेशनसारखं रेस्पिरेशन त्या ठिकाणी कॅरी करतात आणि ATP जनरेशन करतात. म्हणजे त्यांचं असं नाही आहे की स्ट्रिक्टली ऑक्सिजन नकोच आहे. इवन दो इन एन्व्हायरमेंट लो कॉन्सन्ट्रेशन ऑफ ऑक्सिजन इज देअर, they can carry out, they can conduct respiration and they can produce ATP. So they are going to replace the gene required for anaerobic respiration. In case of aerobic respiration, we know that whatever expelled protons are there outside the membrane, they will accumulate and they will create a gradient across the membrane. As I said earlier, that due to flow of electrons and protons, electrons will flow in the electron transport chain, no doubt, but protons will be expelled out. and high concentration of protons will be there outside the membrane. So this will create gradient. High concentration of proton will be outside the membrane, and inside the membrane low concentration of proton will be there. So this is the gradient creation. So these protons try to re-enter. Whatever protons are thrown outside the membrane, जे protons सेल मेम्ब्रेनच्या बाहेर फेकले गेलेले आहेत ते पुन्हा सेलच्या आतल्या साईडला यायचा प्रयत्न करणार. सो दे आर गोइंग टू re-enter. They try to re-enter inside the membrane, but how they are going to re-enter? त्यांना एंटर होण्यासाठी काहीतरी एक लोकेशन किंवा जागा पाहिजे. सो दॅट लोकेशन इज नथिंग बट द साईट फॉर ATPase synthase. So through this organism, they try to enter, that means the protons, they try to enter inside the cell, and here the organism, in this organism, the whatever protons are there, they are going to enter inside the cell. They try to re-enter inside the cell, and this proton enter through the site where ATPase enzyme is located. So there is a conformational change in ATPase unit. ATPase enzyme हे unit आहे. It is made up of different subunits. So there is a conformational change in the structure of ATPase enzyme, and this brings about the synthesis of ATP. Re-enter होत असताना, प्रोटॉन्स re-enter होत असताना कुठल्या साईटमधून, ATPase साईटमधून, त्या ठिकाणी ATPase एन्झाईमच्या स्ट्रक्चरमध्ये किंवा कन्फर्मेशनमध्ये थोडासा चेंज होतो आणि त्यामुळे त्या ठिकाणी ATP सिंथेस होऊ शकतं. सो देर इज अ सिंथेसिस ऑफ ATP बाय रिलीजिंग फ्री एनर्जी. So in this way, whatever protons are expelled outside the membrane, they will create the gradient across the membrane, and these protons try to re-enter inside the cell, and they enter through the site where ATPase enzyme is located. So there is a energy generation due to conformational change in ATPase enzyme unit, and there is a ATP synthesis will take place by releasing the free energy. So this is regarding the mechanism of ATP synthesis in aerobic and anaerobic microorganism. So if any difficulty, you can ask me. I have completed the syllabus of this semester. I have compared metabolism topic. If any difficulty in any query is there, you can ask me. Thank you.